G'day, I'm Dr. Philip Button from the Food Entrepreneurship Academy. Here I am in the Docklands Business District at the bottom end of Collins Street, right at the end where Collins Street, close to where Collins Street meets the Yarra River. And what I want to talk to you about in this video is about controlling microorganisms, controlling their growth. And the principles, briefly, some of the principles behind the different types of control mechanisms. I'm just going to share my screen and take you through a particular slide on that. So here we are. So what I want to go through in this particular slide here, to start off with is basically some definitions, and you can see on the right side, right down the bottom are some definitions. So these are, really important because some people, sometimes interchange them, but they are distinct and they are, quite exacting, really. So disinfection, so what is a disinfectant and what does it mean to disinfect something? So that's the destruction or the removal of vegetative cells. So that means bacteria that are actually viable cells. So it doesn't mean to remove spores, so endospores. And so endospores are a non-vegetative state of a bacterium where you can think about it like a hibernation state. And in that particular case, they're not actively growing, and they're really particularly resistant to a lot of methods for their removal or inactivation. So disinfection doesn't aim to eliminate or remove those. It just eliminates and removes the bacteria that are actually growing, actively growing, or capable of that. And it's actually, mostly used on inanimate objects. So this contrasts to sterilization, which is complete removal. So all viable microorganisms, which is meant to include spores, so that there's nothing capable of actually growing or, actively growing now or in the future. So in terms of spore germination and that sort of thing. And in this case, it is always used on inanimate objects because it's pretty severe, intense, the control or treatment mechanisms. Antiseptics are chemicals which are applied to body surfaces. So destroy or inhibit vegetative pathogens. So pathogens that are actually, capable of growing or are actively growing. And if you put an antiseptic on your skin, then, like an alcohol-based antiseptic, for example, then that's what will inhibit those particular microorganisms that are actually there and capable of active growth. Now, chemotherapy is all about chemicals that are applied internally to kill, which are bactericidal, or inhibit, which are bacteriostatic, growth of organisms, microorganisms, bacteria within the host. In this case, we're talking about microorganisms, but people generally regard or talk about chemotherapy in the context of cancer. But in this case, we're talking about inactivation of bacterial cells. So in this particular slide, we've got different types of control mechanisms: physical agents, chemical agents, and mechanical removal. And what isn't included here is biocontrol, biological control methods, which is much newer concept, really, and certainly one that's not actively practiced, at least in a commercial setting. And if we're talking about biocontrols, we're typically, in this case, I'm particularly talking about bacteriophage biocontrol, so phage biocontrol, which are specific for a particular bacterium. And I'll put a link to that video at the end, which I've done previously, which shows a bit more about that. But with physical agents, you can have heat, which can be dry heat or moist heat. Now, dry heat is most intense, and that is, it has a sterilizing effect, whereas moist heat may or may not be. So, just boiling and pasteurization and those sorts of things have a disinfection, act as a disinfectant because they're not particularly intense. But if you apply them under pressure, so like steam, autoclave, that sort of thing, that's moist heat, but it's under pressure, and so that can have a sterilizing effect. Whereas boiling water doesn't, because it's not at a high enough temperature. Now, radiation can either disinfect or sterilize, depending whether it's ionizing. In the case of X-rays, gamma rays, so we're talking about irradiation, which sterilizes and has can be used for food safety purposes, but non-ionizing radiation, such as UV, can only be used for disinfection, and therefore it can't be used for, by and large, can't really be used for food safety purposes. Now, with chemical agents, we've got gases which can disinfect or sterilize, so like ozonation, ozone, for example, and various liquids, so gaseous, liquid chemicals that can be applied to either animate or inanimate objects, to, in the case of inanimate, disinfect or sterilize, or as just mentioned previously when I went through the definitions, if you're applying it, you've got chemotherapy and you've got antiseptics, depending where you're, what the purpose of that chemical process, chemical application is. And then you've got mechanical removal, such as through filtration, in the case of air, with disinfection, with like HEPA filters or those air filtration mechanisms. And then you've got liquids, where you've got liquid filters with different pore sizes, and you can have pore sizes that are extremely small, which can have a sterilizing effect. So I wanted to cover off ways that bacteria, particular bacteria, can be controlled through physical, chemical, and microbial, mechanical removal. And in another video, which I'll put a link to in a moment, you can also have a look more into biological control, so phage biocontrol. And also just remember the differences between disinfection, sterilization, and then chemotherapy and antiseptics. So that's it for this particular video. And we stop share, and there we go. So yeah, thanks very much for listening. And if you like what we do, like the videos and the types of content that we deliver here, please consider subscribing and check out some of our other videos. In particular, the one I've got up here on phage biocontrol. So thanks very much, and look forward to your company next time. That's all for now. Signing out from the Docklands Business District. Thank you, and see you around. Bye.